Good morning and good afternoon, everyone. Welcome to this Eye on the Future Virtual Fall Summit. We're very happy that all of you could join us today. And I think we have a very exciting program that's featuring our Sepofarsen data and the patient perspectives in clinical trials. So as a formality, during today's presentation, we'll be making forward-looking statements. So please review our SEC filings, which are also available on our website and also for more information. So 7 months ago, we all came together for the EFF 2020 meeting that we did in Paris, which was our in-person meeting. And I think the world looked very different back then. COVID-19 introduced uncertainty into our lives and has impacted us in ways we could not have predicted when we last saw each other. Our connections with family, friends, colleagues, and our broader community was interrupted in meaningful ways. This disruption makes it even more important in the era of COVID to reach out, to share information, perspectives, and to also reaffirm our commitment to each of you, our partners in this journey to finding treatments for inherited retinal diseases. The Eye on the Future Forum was created by ProQR to serve our community of patients, caregivers, advocacy groups, and healthcare professionals. Our goal is to bring each of these virtual groups together in an ecosystem of people invested in curing inherited retinal diseases, understanding the clinical, emotional, and social impact of these diseases, and working together to develop treatments. This Fall Summit represents an opportunity for us to share important new data of our Sepofarsen program in Leber's congenital amaurosis, and to continue to create meaningful dialogue to further our understanding and support of connections. To this end, we've brought together an exciting group of speakers to highlight the work that we've been doing in this space. So first of all, Francesca Diodati is based in Parma, Italy, and lives with Leber's congenital amaurosis type 10. She's been working with the University Hospital of Parma Research and Innovation Unit and is a member of the ProQR Patient and Caregiver Steering Committee, where she acts as an advisor to ProQR. Second, Dr. Ian McDonald is one of our top experts in the inherited retinal diseases field and is currently Professor Emeritus at the Department of Ophthalmology and Visual Sciences at the University of Alberta in Canada. And third, Dr. Aniz Girach is our Chief Medical Officer at ProQR. He's an ophthalmologist and has more than 20 years of experience in the pharmaceutical world, and he holds an honorary professorship at Wills Eye Hospital in Philadelphia. So, to take a look at the inherited retinal disease field, there are more than 2 million people in the world that live with an inherited retinal disease, and this is often affecting children early in their life. These are diseases that lead to blindness in the retina in the back of the eye. Very few of these 2 million people, less than a few thousand, have a treatment available to them. All the other patients have no treatment options and are currently destined to become visually impaired. We believe that our technology can lead to medicines for approximately 25% of the mutations that cause the inherited retinal diseases, and possibly more. So we are committed to building out our platform to develop medicines for these patients in need. To that end, we have built a pipeline of medicines that each target individual mutations that lead to these diseases, together forming a very sizable group of patients that we may be able to help with the medicines that we develop. Our pipeline has been steadily maturing over the last few years. We have our program for Sepofarsen, which is currently in a pivotal study called Illuminate, in which we study Leber's congenital amaurosis type 10. This study is currently enrolling in both Europe and the US. 2 more drug candidates are in a first-in-patient trial. The QR421A program for Usher syndrome and retinitis pigmentosa in the Stellar trial, and the QR1123 program for autosomal dominant retinitis pigmentosa in the Aurora trial. A 4th program for Fuchs endothelial corneal dystrophy will start a clinical trial very soon. And beyond that, we have dozens of earlier stage programs that could enter clinical trials in the years to come. Due to the generalizability of the behavior of the molecules that we are developing, we're developing these drugs as a platform. Molecules that are so similar that the learnings on each molecule can inform the next molecules in the pipeline. That makes us believe that we have a platform with a very scalable opportunity across many of these inherited retinal diseases. So switching gear to the impact of COVID-19. While there was an impact on our clinical trials due to COVID-19, ProQR has an unwavering commitment to bring medicines to patients. Our priority has been to ensure the safety of people participating in our trials, as well as the healthcare professionals who care for those trial participants. We worked with each site on creating a plan to maintain safety and data integrity so that we faced at least the least possible disruption in the trials. Internally, we refocused our efforts on identifying patients and putting infrastructure in place to support patients and sites when a sense of normalcy returned. And we continued this commitment to providing eligible patients access to our trials and to the sites with the resources that they need to deliver the clinical trial care to patients. We're very grateful to the clinical trial sites for working with us through this COVID-19 pandemic and are pleased to see that many of the sites are getting back to normal operation. So for the remainder of this call, I will now turn it over to Aniz Girach. Aniz will start an exciting update on the emerging data from our Sepofarsen clinical trials. I will then proceed into discussions with Dr. Ian McDonald and Francesca Diodati. Aniz, over to you.
Thanks, Daniel. And if we can move to the first slide, please. So I'm going to talk a little bit more about Sepofarsen, which is our antisense oligonucleotide RNA therapy, previously known as QR110, for Leber's congenital amaurosis type 10. And I'm going to also describe to you over the next few slides what trial data we have available and the exciting prospects that we have for the future of potentially finding a therapeutic drug which could be safe and efficacious in this disease area, and in fact in many other disease areas in the inherited retinal disease space. And so this is a very exciting time to be in this area at this stage for clinicians, and I'm sure that Professor McDonald will also echo that when we speak to him a little bit later. So LCA10 is, of course, a blinding disease. There are no treatments out there available. Approximately 2,000 patients are in the Western world who suffer from the specific mutation that we are targeting with Sepofarsen. And our goal ultimately is to either restore the vision or prevent further vision loss in patients who have LCA10 at the moment. The treatment itself is locally administered to the back of the eye through an intravitreal procedure, which is an in-office procedure. And it is anticipated that it's going to be a 6-monthly dosing regimen. We know that Sepofarsen has got some very exciting phase 1/2 top-line data, which I'll share with you in just one second. However, it's important to note that the regulators have also acknowledged the importance of this particular drug and indeed this disease area as an exciting and important area for drug companies like ourselves to explore. Importantly, Sepofarsen is only the second drug to have been given European Medicines Agency PRIME designation in the ophthalmic space. And so we're very excited about that. And like Daniel mentioned, we have an ongoing phase 2/3 Illuminate trial, which we're actively recruiting for at this stage, and we'll share more information about that in a later slide. If we can go to the next slide. This slide summarizes all of the different trials that are ongoing at the moment in Sepofarsen and those that are completed. So for example, I will share with you some of the early data that we've got already from the phase 1/2 trial, which is completed. There is an ongoing extension of that phase 1/2 trial called the InSight trial, and that I'll share with you some early preliminary data that's coming out, which is very exciting for us. We'll also share with you the overall trial design for the ongoing and actively recruiting Illuminate trial. And you can find more information on that Illuminate trial on a specific website called lcastudy.com. And we also have an ongoing validation study for the mobility course, which is one of the many endpoints that we could study in patients with LCA10. And we also have plans to be able to start a pediatric study in the future into 2021 and beyond. So the phase 1/2 study, that was the first in human study that was performed a few years ago, and that just finished at the end of last year, was an open label study looking at LCA10 patients with different visual acuities at baseline, and indeed included both adults and pediatric subjects as well. 2 different doses were explored. The 160 micrograms loading and 80 micrograms maintenance dose was the lower of the 2 doses, and there was a higher dose, which was 320 micrograms loading and 160 micrograms maintenance. We will focus more on the target registration dose, which is the 160 micrograms loading and 80 micrograms dose as we move forward. And indeed, we found that the benefit risk of that particular dose was much more favorable compared to the higher dose that we had observed. We also have now rolled over any eligible patients into an extension trial. The extension trial is called the INSIGHT trial. And I will share with you also a 3-month interim analysis out of the INSIGHT trial in the subsequent slides to come. And so if we move to the next slide, this slide summarizes the efficacy data that we have available from the phase 1/2 study in the target registration dose of 160 and 80 micrograms. This was, remember, a phase 1/2 study, so it's a first-in-human study where one eye was treated and the fellow eye was untreated and used as a control. The main summary outcomes you can see here on this chart are best corrected visual acuity outcomes on the left, a full field stimulus threshold outcomes in the middle, and the mobility course outcomes on the right-hand side of the chart. The BCVA are probably the most striking of the results, where in the green line, which is the first eye treated, you can see importantly a clinically significant and meaningful change, improvement in vision of about equivalent of 9 lines on the ETDRS chart in the treated eyes compared to the fellow control eyes, where, as expected, they are probably within the noise of the test of the assay. This is highly clinically significant, albeit that it's in a small number of patients. And of course, this needs further validation in a larger trial, such as the phase 2/3 trial that's ongoing. Importantly, this best corrected visual acuity was supported by other measures of outcome measures, which were also important to note. And the one that I'd like to turn to next is the full field stimulus threshold readout, where we tested both red and blue lights. And in the solid lines, you can see an important and clinically significant change in the mean change from baseline, where a downward motion, just like the BCVA graph, is an indication of efficacy and improvement. And you'll see there that it's in the treated eyes, which is the solid lines, there is a greater treatment effect, a greater benefit seen compared to the untreated eyes, which is in the lighter lines. Further support of this is also given by the mobility course assessments, where in the green line, you can see the treated eye responding by a series of mobility levels change from baseline. Here, an upward movement is beneficial, and the yellow lines are the corresponding levels in the untreated eye. So importantly, as I look at the 3 sets of outcomes there, the best corrected visual acuity is, of course, a registrable endpoint. And like we said earlier, that albeit with the caveat that this is a first-in-human study in small numbers of patients, it's highly encouraging to see that effect, a clinically significant effect in the BCVA, and also supported by other outcome measures as well. And so if we move to the next slide, please. Given the potentially beneficial effect of the phase 1/2 study, at the end of that study, all patients had an opportunity to roll over into an extension study. And indeed, 4 patients have been treated so far in the long-term extension study, and these are summarized in the chart that's on this graph at the moment. We've included both the BCVA responses and the FST responses, both red and blue, and an upward movement is an improvement. And you'll see that the participant number 2, participant P3, participant P11, and participant P1, all of these participants showed a beneficial effect on both BCVA and or FST, both in red and blue. Participant P1 started off at light perception at baseline and unfortunately did not have any improvement on the BCVA. However, you can see from the FST response there, both in red and blue, that there was a significant improvement in the patient's light sensitivity or retinal sensitivity as measured by FST in both red and blue FST measures. So these were the first eyes that were treated and the responses that you can see there in those patients. If you then click to the next slide, the yellow bars now indicate the second eye treatments when the second eyes were eligible as part of this extension study. And importantly, it's worthy of note that the second eyes behave in a very similar way to the first eyes, in that the responses, both in BCVA, if there is appropriate, and or FST, both red and blue, are very similar and correspondingly favorable in all of these patients that have been treated so far. And if we then move to the next slide, please. I'd like to just maybe highlight one of those patients, which was participant P3. And this is an illustration of what the best corrected visual acuity improvement means in real life for that patient, for example. This patient started off with off-the-chart vision, which means that this patient could not read any chart letters at all, and was in fact very poor in their vision down to hand motion level. After 3 months of treatment with the second eye, this patient improved to reading the lines on the eye chart, and in fact carried on improving right out to month 9 with a very significant improvement in vision, as depicted in the series of pictures that you can see there. Whereas the left-hand side is the baseline evaluation, where the patient could barely see the chair or any charts on the wall. And you might be able to perceive that the month 3 evaluation, and in fact, even the month 9, allows the patient to be able to see objects in the room much more clearly, and including even different lines on an eye chart that could be up on the wall. So this is just an illustration and gives you an idea of how a patient who would be legally blind to start off with could potentially be navigating freely, watching TV, and being much more independent as a result of the treatment. And of course, we hope that a lot more patients have the same kind of benefit in future trials as well. And if we move to the next slide. Now, this data out of the phase 1/2 has significantly informed us of our ongoing, actively recruiting phase 2/3 trial, and indeed allowed us to be able to target a population of patients with a best corrected visual acuity of hand motion or better, where we saw the most amount of benefit. However, it is important to know that the benefit that this drug had on these patients was noted across the board. That is to say, all patients benefited in one format of an endpoint or another. And I'll illustrate that in these subsequent slides. And so if you move to the next slide, please. Those patients on a logMAR scale who have light perception only that were included in this phase 1/2 study, there was one patient who had an outstanding improvement in vision. The downwards motion is the direction of improvement, and this patient had almost a 26-line improvement, equivalent improvement on the ETDRS chart in the first eye, and a similar level of improvement in the fellow eye, the originally control eye, and when that second eye was treated as part of the extension study. The other patients who had light perception seemingly did not improve on best corrected visual acuity. But as you'll see in a later slide, they actually also showed improvement on FST. All of the other patients recruited in this phase 1/2 study had a visual acuity of hand motion or better. And it's evident that all of these patients had an improvement in vision. And the majority of these had an improvement in vision, which was clinically significant, even at the regulatory threshold level. And so this phase 3 population that we're studying in the Illuminate study gives us great confidence in the potential outcome of that study if these study results were to be replicated, in that the mean change in vision in those patients who had hand motion or better out of the phase 1/2 study had almost a 5-line improvement as a result of the change that occurred with the treatment. Now, if we then focus back again to the patients who had light perception, and especially those patients who seemingly did not have the visual acuity improvement, that is to say patient number 1, number 6, number 9, and number 10, If you move to the next slide, please. The full field stimulus threshold responses, that is to say, those patients, the ability for those patients to respond to light sensitivity measures is given in this chart. And it's very evident that there are improvements across the board in all of these patients in both red and blue formats of sensitivity measurements. And this is the change from baseline that you note here. So even though these patients may have had light perception at baseline and were not able to improve on visual acuity measurements, which are perhaps sometimes rather crude assessments of the overall vision, it is evident that on more sensitive measurements, such as FST, that they do show a response, a favorable response, in both the first treated eye and then also in the second treated eyes as well. And so that's very encouraging for us to know that the applicability of this drug could be much more widespread than initially thought, and that it's not just the visual acuity that is a measure of improvement, but actually other endpoints could also pick up improvements in light sensitivity or perhaps other measures, which could be equally important to patients sometimes. And so this is being studied in the next slide. This is being studied in our phase 2/3 pivotal trial called the Illuminate study. And this is, of course, actively enrolling. And this is a 3-arm trial, 2 treatment arms, looking at the target registration dose of 160 loading and 80 micrograms maintenance, as well as a lower dose of 80 micrograms and 40 micrograms maintenance with a sham procedure. And this is a very well-known trial design for regulatory purposes. And it's a 24-month study with a 12-month primary endpoint readout and 6 monthly injections, both in the treated eye, first of all, and then after the 12-month time point, the untreated eyes can also get treated as well, including the patients who are allocated to the sham procedure in the first 12-month time point, where they would be eligible to cross over into one of the treatment arms after the 12-month time point. So this is a classical double-masked randomized controlled study. We have sites globally. So we have sites in the US and ex-US, encompassing the EU and also Canada and Latin America. We have patients who would be eligible to enter the study who are 8 years and above and who fulfill a criteria on visual acuity of hand motion or better. We will look at visual acuity as our primary endpoint and, of course, assess a number of other key secondary endpoints as well. And I'd like to stress that this trial is actively enrolling, and therefore, if anybody is interested in participating in this trial, you can find out more information from our website, ProQR.com, or indeed the specific website for this particular study called lcastudy.com, or talk to your ophthalmic caregiver who will be able to have more information for you to be able to provide to you. And they would be also be in a better position to be able to assess your eligibility for this particular trial. We have experts across the world who are participating in this trial, and therefore there won't be an expert too far away for you to be referred to be able to assess the eligibility for being included in this particular trial. And if we then move to the next slide, please. So our future plans for LCA 10 in particular, and then also in LCA, are that we do anticipate starting a pediatric study in patients who are younger than 8 years of age next year or as soon as possible. This will be a 2-year study looking at patients who have visual acuity of light perception or better. That is to say, the ability to be able to tell the difference between light and dark. We anticipate this study to be across multiple countries. And then we have the ongoing studies, both the InSight and the Illuminate study, which will continue to participate and actively treat patients, even over the long run, beyond the current trial durations, and right out to when, hopefully, the treatment would be available commercially as an approved therapy in the future, we hope. And so I'd like to just pause there. This is the series of slides that I wanted to present. I'm sure there'll be questions on this in the question and answer session, which is what we'll turn to next. And so I'd like to introduce Jessica Ibbotson, who is our head of operations at ProQR, and she will hopefully shepherd us through the live Q&A with Dr. McDonald and myself.
Yep, thanks, Aniz, for that. So this is the panel portion of our agenda. This will be a 25-minute Q&A, and then we'll move to our second panel. So thank you, Dr. McDonald, for joining us as well. Maybe I can start with both of you as an introduction to just share your experience and what is your experience in the field of IRD and maybe specifically LCA10. So maybe Dr. McDonald, if I could ask you that question first.
Sure. Thanks very much, Jessica, for asking me to join you in this very interesting discussion. I was reflecting upon how I started in this field. In the early 1980s, we didn't know which genes were actually involved in some of these disorders. And so with the Human Genome Project, there's been such discovery to be able to understand these inherited retinal diseases, which I've worked on since the early 80s. And to see the advance from gene discovery to then potential treatments is remarkable. And this example is quite encouraging for the entire field. So I'm an ophthalmologist and a clinical geneticist by training. The two elements of medicine are particularly helpful in me understanding the potential of these treatments for patients and families.
Thanks, Dr. McDonald. And Aniz, do you want to share your experience with IRDs and LCA10?
Sure, and you know, my background is not as extensive as Professor McDonald's. I'm an ophthalmologist, a clinical ophthalmologist, as a retina specialist, and of course, I've managed patients with IRDs over the last 30 years, and particularly over the last 10 years, and especially as part of the industry, I've focused much more on the inherited retinal disease areas, first through gene therapy, and now through RNA therapy. And so, you know, really excited, like Professor McDonald is, to be in this area, because I think at last, not only have we found some of the causative genes of these blinding diseases, but at last we're now beginning to unearth some of the treatments that could potentially help these patients and hopefully prevent blindness in the future.
Thank you. And then maybe Dr. McDonald, just kind of following on the data and what you were alluding to in your introduction there. There are many patients that may be on the call or patient advocacy groups who interact with patients on a daily basis. So how do you put the Sepofarsen efficacy data into perspective for somebody that has LCA10 or a caregiver of someone with LCA10?
Well, I'm very optimistic. This is a potential treatment that we can see that will help patients with LCA10. LCA is a very complex disorder. There are many genes. This particular genetic mutation that is amenable to RNA therapy has the Sepofarsen product is, I think, an outstanding success so far. So I'm very positive in my discussion with patients about the potential for this. I have the privilege of seeing two patients in my practice, a brother who had initially undertaken this experiment and received significant benefit from it, and now his sister enrolling, seeing the benefits in the brother, and having them relate to me the benefits that they've experienced, not how many letters, but just their own perceptions of how much they have benefited from this experience of enrolling in a clinical trial.
Great, thank you. And you mentioned RNA therapy, and Aniz ProQR is obviously focused on RNA therapies. So maybe you can just talk a little bit about ProQR's vision for treating people with IRDs with RNA therapy.
Well, as Daniel said at the beginning, there are many disorders, human disorders, affecting the retina that may be amenable to RNA-based therapies. And having worked in this field, looking at the other products that are available, for example, viral mediated gene replacement, and the difficulties involved in the delivery mechanism and also the trigger of immune responses, I feel that for many individuals, the RNA-based therapies have a better safety profile. And so for the patient, knowing that is quite reassuring. So that's my perspective, and it I would say, probably matches many others in this field, that there's great potential for this approach for a broad number of inherited retinal disorders.
Great. And Dr. Girach, anything you want to add to that?
Yeah, just to layer on top of what Professor McDonald was saying, that as a company, we're very excited to be leading at the forefront of this technology. Our aim, like Daniel mentioned earlier, is that we want to at least be able to provide a safe and effective treatment for as many of these 2 million patients who suffer from inherited retinal diseases ultimately. And we think that our technology can at least capture 25% of those patients and help at least 25% of those patients somehow either restore their vision or even maintain their vision. And so we're working night and day frantically around this to be able to make that happen. We're also very excited at our participation in ensuring that the genetic screening processes are also going in the right way. And we've partnered with the Foundation of Fighting Blindness and the MyRetina Tracker to ensure that there's at least free provision of genetic screening in the US. And so we believe that that's quite an important step in ensuring that patients get diagnosed appropriately. And hopefully with the multitude of treatments coming their way, this would be at least a better future for most of them.
And there are just a few questions that have come through related to genetic testing. So just a reminder to anyone watching, please submit your questions via the chat function, and we'll make sure that we get to as many of those as possible. So one of the questions was related to which registries, not necessarily MyRetina Tracker, I think many people in the US may be familiar with it, but which registries would ProQR be willing to collaborate with to inform, educate, and allow equal access to therapies? And maybe I can start to answer that and send it over to you, Aniz, because it also follows on another question about equal access. So it's obviously something on people's minds. I can just speak generally about what we've been doing in clinical operations and across the organization to reach as many people as possible. So, this is obviously a very rare disease, and it's not always well known where all of these patients are because genetic testing is not consistent globally. And in particular, we see that definitely in the US and maybe a little bit less in Europe. But we do try to collaborate with as many registries as possible that may have patients with LCA10. We do offer, as part of the Illuminate studies and all of our studies, genetic testing if the patients are deemed eligible on the main criteria by the physicians. So I would say just generally our approach is to be as inclusive as possible, to reach out really globally. And we have reached out to even countries that are not participating in the trial to try to find LCA10 patients so that they can be educated about the opportunity for the clinical trial, that ophthalmologists globally are educated about what we're doing at ProQR, and try to ensure that equal access to patients. And Aniz, I don't know if you want to add anything on that.
No, I think you mentioned that very nicely. I think the important thing also is that we have a series of sites and countries that were already opened a site for inclusion in the Illuminate trial. And like you mentioned, this does not exclude other patients from participating in that trial. There are various examples that we have already in our ongoing trials where patients have been referred cross-border in order to be part of a particular trial. And therefore, it should not stop anybody from finding out more about whether they'd be interested to participate in a trial and educate themselves and ask their treating physicians if they would be appropriate candidates for this. And then the treated physician or the patients can certainly reach out to us at any stage or any of the existing sites that are available on our website as well.
Yeah, and that's an important point. And just to be kind of crystal clear for any patients that may be listening, just because we're in certain countries with the clinical trial does not mean, if you're in Poland that you can't participate in our clinical trial. We make provision to ensure that any eligible patient is matched up with a site as close to them as possible. And so just want to make that clear. And just kind of maybe expanding on that, Aniz, one of the questions that's come through is, have you thought of expanding your phase 3 trials beyond the current countries? Why not? And just for everyone's interest, I can tell you that the current Illuminate sites are in the United Kingdom, Belgium, Netherlands, Germany, France, US, Canada, and Brazil.
Yeah, it's an important question. We're currently having at least 20 sites across the globe that are participating in this trial. Of course, it's always a challenge to open up new sites because it takes time, energy, money, and resources on the ground to be able to open up new sites. And of course, it requires patients to be there on the ground also that are eligible. So we're always open to new sites opening, but of course there's also a time component to this that we don't want to delay the overall progress of the Illuminate study, and therefore we'd like the Illuminate study enrolled as soon as possible so that ultimately we can find out with some level of surety whether the Sepofarsen drug is safe and efficacious for patients. So it's always a balancing act, but I feel as though there are certainly 20 sites already open in various geographies, and so hopefully there'll be ample opportunity for any patients that may be interested in taking part from various other parts of the world to be able to get to the nearest center that could be participating here.
Great. And then maybe a question for both of you, since you have come at this from different angles. Why does it take so long after the discovery of a potential medicine before it is actually available to patients? The COVID-19 vaccine program shows us drug development could be much faster. So again, I think this speaks to this theme of access and really getting treatments to patients as quickly as possible. So maybe Dr. McDonald, if I could start with you.
Sure, thanks, Jessica. So the example of COVID and herd immunity really is different in a sense. So you're trying to develop an immune therapy based on vaccination and to an entire herd. And this is an infectious disease, not a degenerative disease. And so there's a compelling reason to use any technique, a pharmacological technique or a vaccine. There's an urgency around it. Not that there isn't an urgency around an inherited retinal disease, but we're talking about a treatment here that is going to be lifetime versus a one-shot immunization, and maybe it works, maybe it doesn't work. There's a lot of dialogue out there about COVID right now, and so we can, let's say, put that aside for the moment and talk about inherited retinal diseases and why is it taking so long? Well, let's say the first ones took a long time, but one can see that the advances are occurring more rapidly now. The genes are now largely known. There are a few that are becoming known now that we hadn't known before, but they're very rare. We can see that in some of these, they may be amenable to RNA therapies. And this wonderful example of Sepofarsen is right in front of us as a almost fast track. The patients are still within clinical trials, but I would imagine that the this data very shortly can be presented to the EMA in Europe or FDA and say, we would like to be offering this as a treatment. So, and a treatment is outside of the clinical trial, but the supplier, the manufacturer, ProQR is still under the obligation to provide information on the safety and efficacy of that drug in the marketplace. That the regulatory agencies want to do that. But there's that transition in between a clinical trial and a treatment that takes some time because the regulator wants to put materials into the clinical space as treatments. They're not there as a barrier. They're there to help that process. But all of this data is going to be convincing. It's going to be the basis on which the Food and Drug Administration, for example, in the US says, yes, you're ready to go. And you can see in this product, although this has taken a fair amount of time with basic science, the genetics of the disorder, the use of models to say, yes, if you add an RNA, it will allow you to provide a normal protein, what that has in the model as an effect, what it has in the human as an effect. All of that goes into the binder that goes off to the government. It may not be a binder anymore. It may be electronic. But I sense that this product will become a treatment very shortly. I'm not obviously aware of how long shortly really means, but it's not going to be the extended course that some of these treatments have had in the past.
Thank you. And Dr. Girach, anything you'd like to add to that?
No, I think Ian has pretty much mentioned all of the important points. I guess also just elaborating on what he mentioned about Sepofarsen. Sepofarsen actually is a very nice example of a fast-tracked program. In that using our proprietary OptiCup models, we've been able to shorten the time span between drug discovery and actually getting into clinical trials, into pivotal phase 2-3 clinical trials also, and hopefully in the future an approval of the drug. And so this is a very good example of that based on our preclinical OptiCup models that we've developed in-house. I guess another point just to layer on top of what Professor McDonald was saying is that when you compare with COVID-19, of course, there was a lot of talk, hype, money being pumped into research into that field, and appropriately so, of course. That's causes mortality and significant morbidity. But I think that if the same amount of awareness and also funding was available for research into the inherited retinal disease spaces, then I think it would make a big difference to potentially finding treatments much earlier in these spaces as well. So we look forward to that day, one day when our governments are also looking at degenerative diseases in the same way as other diseases that are taking more prime time on the TV at this stage. And like I said, that's with all due respect to COVID-19 and with all the harmful effects that it does cause as well. We believe that we want to be able to invest significantly in terms of resources and money into all of these significant diseases that cause morbidity and mortality, of course.
Great, thank you. And another question has come in about Sepofarsen. The question is, will Sepofarsen improve peripheral vision, tunnel vision as well? And this may be a good opportunity to talk about LCA10 versus other types of IRDs. So maybe, Aniz, if you want to start related to will Sepofarsen improve peripheral vision, and then maybe Dr. McDonald, if you could speak a little bit about LCA10 versus some other IRDs, types of IRDs.
Yeah, and so Jessica, so far, it's early days in terms of the treatment and the benefit that we can see. We've seen that the beneficial effect of Sepofarsen is in central vision with best correct visual acuity. We've also seen that in the periphery of the retina, where the mixture of rod and cone photoreceptors exists, there is an improvement in the sensitivity, in the light sensitivity of these patients. But we don't know at what extent that ends, whether it's just the improvement in light sensitivity or whether indeed there's any improvement in meaningful vision that comes in that peripheral vision. And hopefully the future trials are going to be able to elaborate on that. But I think that it's important to also understand the underlying disease, the LCA10, and how that behaves. And maybe, Professor McDonald, you can just elaborate on that in terms of your experience of the natural history of the disease.
Thank you very much, Aniz. I think you captured some of the thoughts that are very important, that the distinction in between one disease and the other, and how the initial clinical trials enroll perhaps patients who have over the years lost a significant amount of their visual function, and function being reflected also in the anatomical changes in the back of the eye. And so let's talk just briefly about the patients in the Sepofarsen trial that had only light perception, and to know that those individuals could improve their visual acuity from light perception to being able to see better is a remarkable feat, because it suggests that the threshold for improvement in vision doesn't need to be set at a certain level. It may be very, very broad. So let's think about that concept first, but let's also consider that these were early trial results that were quite positive, and then you can broaden the scope of the application to patients who are younger. As you mentioned, down to the age of 8, these individuals would potentially not have lost as much visual function and not as much visual structure within the retina. And so in their lifetime, they're probably going to benefit significantly by having that treatment at a very early stage. And that treatment may be reflected in not only broadening their visual field, that's possible, but maintaining a larger visual field. For the inherited retinal disorders, there's two major aspects that we think about constantly. Visual acuity, so in order to see the human face, read, but visual field in order to navigate. And the two are in concert. The anatomy of the retina is such that the cones are the predominant photoreceptor in the central retina, and it tends to be preserved throughout many of the retinal dystrophies, the cone function that is. But the rods are dominant in the peripheral part of the retina, away from the macula. Those, when they degenerate, result in the loss of visual field. This particular approach with Sepofarsen is not targeting just the rods or just the cones. It is agnostic, if you can say that, or Catholic. It's going to affect the cells throughout the retina and improve their function. And so I'm certain, I feel that once more individuals are included in the clinical trial, we'll see that there are meaningful, and that's an important adjective, meaningful changes that are occurring to the patient, and they'll be reporting that, gosh, I can navigate out in public much better than I did before. For the one individual within the clinical trial that I know personally, his visual acuity has improved significantly. His visual field is still not as good as he would like it to be, but with scanning, he can certainly navigate better. So I shouldn't hog the stage here. I hope that gives you a little bit of a perspective that might help address that listener question.
Yeah, absolutely. And I think, one thing that you touched on, which maybe I'll ask Dr. Girach to expand on, was the benefit. And one of the questions that has come through is, will Sepofarsen benefit patients with light perception? So Dr. Girach, based on the totality of the data that we have so far and what you know of the disease and the potential for Sepofarsen, maybe you could further expand on that question.
Yeah, that's an important question because, of course, many patients with LCA10 will have very poor vision, down to perhaps light perception or even worse. And you may remember that I showed some slides which covered some data on 5 patients in our earlier trials who had light perception that entered into that first trial. And out of the 5 patients, one of those patients had a great response to visual acuity, but actually importantly to me, and to many of the experts in the field, that all of the patients with light perception improved on the light sensitivity measure, the FST measure. Now, that doesn't mean to say that, of course, all of the patients out there will improve. It just means that these particular patients who entered the study improved. But that gives us an important signal that patients who might have light perception may not be beyond the point of recovery, and that there may be treatments available that may benefit them. And so as we include more patients in our clinical trials and we learn more about the drug and more about the safety and efficacy of the drug, we'll be able to answer that question in a much more definitive way in the future.
Mm-hmm.
Great, thanks for that. So we're just at time for this portion of the Q&A, so thank you both, and you'll continue on with us as we move to our next panel discussion, which I'll ask Francesca to join us. And Francesca, if you can Alt-V and Alt-A. Yep, we see you perfectly.
Hi,
and
hello.
We hear you great. So I'd like to welcome Francesca Diodati. Francesca is based in Parma, Italy, and she works at the University Hospital of Parma and has LCA 10. So thanks, Francesca, for joining us.
Thank you. Thank you very much. I'm really honored to be here, to be on board in this exciting forum and in general with ProQR.
Great. Well, we're honored to have you here as well. Yeah. So thank you. I'd like to just remind everyone once again, if you have any questions, feel free to send them through the chat function and we will get to them. So this is, maybe Francesca, we'll start with you. As someone living with LCA10, kind of what is your perspective on the Sepofarsen data?
Well, they're very exciting, extremely exciting. First of all, because there are data on a study on LCA, which, it was unthinkable only a few years ago. And second, and most importantly, because it works. So, I mean, it's wonderful. And the side effects are really minor. I mean, the only big one might be cataracts, but, cataracts sometimes occur anyway with LCA. I mean, I have cataract in one eye, so you might need to remove it anyway at some point in your life. So it's not a big deal. And I think, the data are very encouraging. And, just the fact that we're talking about, I'm 49 years old. So just the fact that we're talking about results on a study, on a clinical trial on this disease is fabulous. And the fact that it works is wonderful. And I was a bit worried when I saw the first results about light perception, about patients with light perception, especially in the first the interim results. I mean, the results that were shared last year. But then, seeing these results and the fact that light sensitivity improves, I mean, it's astounding because you can do so many things just by seeing light versus no light. So I figure if I can see more light, I can navigate better. And also, I think I'm convinced that if you don't do rehabilitation, just like, patients who have a stroke, for example, and they have to learn to use or reuse a part of the brain, people who only have light perception, and especially those who are never seen before, more than that, and it's, I think, the majority of people with CEP 290, they should be trained to use their brain. And I actually would like to ask Dr. McDonald, maybe if you can tell us his opinion about that as an expert, if he thinks that rehabilitation might help and might play a role in this.
Francesca, it's very nice to meet you again.
Thank you.
It's a wonderful experience to be able to link across the world
yeah.
and understand other people's experiences. So I worked very closely, and I was a board member of the Canadian National Institute for the Blind. And they are much like Lighthouse and many of the other organizations throughout all countries in the world that try to rehabilitate individuals with blindness, important partners with caregivers. I have a finite amount of time that I can spend with the patient, and I really depend on these institutions to help. And rehabilitation, I think, is a key in the whole process. And we could spend more than they've allotted time talking about this. It's very important. It's something I hadn't considered, and it would be something that I think I should think about if I'm seeing the patients, that the rehabilitation specialist within a low vision service see pre and post and say, okay, well, how are we going to adapt to this? What new training modalities are we going to undertake with orientation and mobility training? Maybe we can put that cane away.
Because the fact is that someone who has never seen before has no idea what it means to see. So even if the retinas are fixed, and Sepofarsen can do that, and it's great, it might be that, it's not about opening your eyes and seeing. It's just, I think we need to learn what it means, and the brain must learn. So that might play a role.
Absolutely.
People with light perception.
The brain has all sorts of connections that need to be re-stimulated.
And it can. So there might be more improvement there.
Yeah, the visual cortex is just waiting for the.
Yeah, it is.
And I think that's actually an interesting perspective, Francesca. As we talk to patients and as we hear about physicians speaking to patients and other healthcare professionals, I think one of, the I don't know if it's a concern or at least a consideration is if you go from very limited sight to more sight and you've spent your whole life adjusting to kind of what your visual experience is at that moment, and then it gets better, you kind of have to relearn how to do many things because as you mentioned. So, I think it's been a learning for us as well to really understand. It could be a scary prospect if you've spent your entire life at a certain visual experience to get some sight back. You really do need, in some senses, to potentially relearn things, and it's an adjustment for sure.
Might be scary for some people. I've heard of people who would never take part in a trial, but for others, it might be like an adventure. So it's a way to learn new things. I mean, we are already independent like this without sight. So if we get sight, I think in my opinion, it can only get better.
Right. Agree. And I think one thing, Francesca, you mentioned, and I think it's a good topic for this conversation and for everyone listening. So as you mentioned, clinical trials in LCA10 are relatively new and offer a choice that people with LCA10 haven't previously had. And because each of you kind of come from a different perspective, and Francesca, maybe I'll start with you. How do you find out about trials? It is so new, and you may have never searched for trials before. So how is it that you go about learning about potential trials?
Well, I'm in a privileged position because I also work in research, so I know about use PubMed and clinicaltrials.gov, and I searched the internet, and so I knew about Sepofarsen long before it was called Sepofarsen, and it was in the preclinical stage, and I had emailed the researchers who were working on it, and I had emailed ProQR so often, but usually that's not the case. Not everybody is knowledgeable about research and about the resources. Of course, I mean, anybody is knowledgeable about their kind of job, their kind of work. So I think it's very important that we have the internet, of course, plays a major role now. So sites that are built specifically for patients are very important, because people go on the web and they search, keywords, and they find web pages which should be written in a clear way so that they understand, with contact information where they can, they can write and have more information. Because I think the clinician should be the primary source of information, but unfortunately, it hasn't been my experience. I mean, all my life, there have been a few who have tried, but usually they don't know. I mean, I was the one who explained to my eye doctor what I had and what was about out there, what kind of strategies were being studied out there. So it's kind of hard. I know it's a rare disease and there's so many diseases, and eye doctors, if they're not researchers, don't know. They cannot be expected to know, but maybe they should try to be a little more active in finding out information, or link the patient to experts. Maybe they're doing it more now.
And so, I think that's interesting. And Dr. McDonald, as a clinician, how do you stay informed of clinical trials, and what does a discussion look like with a patient that may have a disease for which there is a clinical trial?
Well, there are many ways of trying to link to clinical trials. One important resource is a website called clinicaltrials.gov, and there's a box that you can type in, for example, Leber's congenital amaurosis, and it will provide a listing of all of the different trials that are current and recruiting, and so that's an important worldwide resource. That said, I must say it becomes quite a challenge to try to address the question from the patient. So it would, let's say Francesca might have asked her physician, what disease do I have? Because I'm not seeing well. I think I have a retinal dystrophy, and can I link to this clinical trial? So then the process starts at that question, and then undertaking the genetic analysis to say what particular disorder underlies the inherited retinal disease or disorder, whatever you want to call it, and then looking up the data that's available on the web, but then sorting through the data and seeing, well, is Francesca eligible for this? And I have the opportunity to work with a genetic counselor who's very helpful in trying to link the information that's currently available, and then have a discussion either in person with the patient and the family, or have a telephone conversation. The web is okay, but as you know, it's very static. There may be there may not be somebody standing by to address your questions, as there are today. So I'll turn off my prologue, and but it is a challenge for all of us. So I'll leave it there, and I'm sure that you have other ways of.
Yeah, and I think that's one reason, and I can speak a little bit on behalf of ProQR, and Aniz, if you want to add anything, feel free to go ahead. As we think about, talking about this ecosystem, there's a lot of push and pull. So Francesca, as someone with LCA10, is trying to pull information out, and Dr. McDonald, you might be trying to pull information in for yourself as well. And we're trying to push out information. So trying to find the right audience, the right mediums to be able to share information. Reaching out to, obviously, ophthalmologists is easy, kind of an easy way to think about who patients that see ophthalmologists, but we also know with LCA10, that they may not regularly see ophthalmologists. So we try to create relationships with patient advocacy organizations, with low vision specialists, with optometrists, and send them information about what we do in case they're seeing those patients as well. So I think there's not one answer about how to do this most effectively, that we're all trying to figure out the best way to give information without information overload, but making sure that people have access. And I think, you know, even kind of a panel discussion like this, we're lucky enough to have some patients that I know have joined to be able to share experiences and information about how we push out information, how you may search for information. But, you know, we're a learning organization. Dr. McDonald, I know you're also a learning organization. So we're always looking for feedback on how to do it better and most effectively. But we do think the ecosystem approach of kind of reaching out to all different groups is the best. And that way we can target as many people living with certain IRDs as possible, so that, you know, we can capture them wherever in the ecosystem they're going, to at least educate them about trials that are happening. Aniz, I don't know if you wanted to add anything.
No, I think you've captured most of the important bits, and I think the three of you have captured all of the important elements here.
Okay, great. And we have a few questions that are coming in, so I want to get to those. We do have someone that has said the AAPOS website, there's a dedicated patient tab, and genetics committee is under that tab. This resource is for patients and doctors related, I assume, to trials. So thank you. It looks like Dr. Girach, Iowa, has provided that information. There is a question. Is there equal transfection to the entire retina? For example, photoreceptors, rods, and cones. So, Aniz, is that a question?
Yes, I can certainly tackle that, and also I'll ask Prof. McDonald to also complement that. So one of the advantages of RNA therapy given through the intravitreal route is that actually it allows you to get the drug across the entirety of the retina. And that's potentially an advantage over some of the gene therapies, which sometimes have to focus in on the central macular zone only, just because of the technologies available at the moment. So the theory, at least behind administering a drug intravitreally, is that the vitreous interfaces the whole of the retina, and you can potentially get access to the whole of the retina. We know that, as Professor McDonald was mentioning, that the central area is principally cones, and the peripheral areas are comprised of rods. And so theoretically, at least, you would get a level of transfection that happens across all of these different type of photoreceptors. And we see that also in some of the results, the early results that are coming in. The central area of photoreceptors are responding to BCVA, best corrected visual acuity, but also the peripheral rod photoreceptors may be responding to the retinal sensitivity improvements that we've seen so far. So at least these early data seem to show that both type of photoreceptors seem to be responding. And, you know, I think that the jury's out in terms of the degree of improvements that we will see across both of these type of photoreceptors at this stage. Professor McDonald, I don't know whether you want to add anything to that?
No, that's excellent. Thank you.
Another question that has come in confirming a presumption: if people with just light perception are having vision improvement, doesn't that imply that they have photoreceptors that are still alive but have stopped functioning, and therefore Sepofarsen restores their function? So, Aniz?
Yes, and I'd like to tackle that first, if I may. I think that that is true. That's at least the theory behind why we see an improvement in vision rather than just stabilization of vision. We think that at the process of photoreceptors, and as they die, they go through a quiescent phase, a dormant phase, where they're not dead yet, but they're malfunctioning. They've got reduced function. And with the appropriate stimulus through a drug vehicle like Sepofarsen, it can actually re-energize the cells to be able to function more optimally. And therefore, that improves the overall function of the retina, and that improves the overall function of the vision as well. And that's how we think that the vision improves at this stage. And indeed, in many there are many examples of this in the literature, in the ophthalmic literature, of where patients' cells improved function, and that led to an overall improvement in vision as well. And again, I want to just invite Professor McDonald to add anything else that might be relevant here.
Well, thank you very much, Aniz. I thought it'd be helpful maybe for the audience to recognize that the rigor that goes into presenting the material to the audience of the FDA or EMA, that they're looking for scientific evidence of improvement. And so this final scotopic or full field scotopic threshold test is a very important test beyond visual acuity and the patient-reported outcomes of benefit. They're wanting to say, well, where's the hard data that's showing that there's an effect, that there's a benefit? And this particular test is invaluable in that sense. It's showing that there is a functional improvement in vision, and the function that is improving reflects a structural change, likely at the level of the retina itself and the photoreceptors. And so we're suggesting that by the improvement in function, there's a structural improvement that's reflected in the photoreceptors, and we have a good marriage of information that's going forward to the regulators who are scrutinizing this particular drug.
Yeah, and I was going to add also, Professor McDonald, that we talk about improvements in vision here. Usually, that's the rarity, and I'd like to also invite Francesca to also opine on this as well. That typically patients, in my experience, limited experience, who have inherited retinal diseases, are actually quite happy if you can even maintain the vision at the same level, never mind showing an improvement. And I don't know, Professor McDonald and Francesca, if either of you can have any insights on that and add your opinions on this.
Should I go first? No. Well, I don't know. I think so. I would think so. Since I only have light perception, I would like to improve it. But if somebody told me you could lose that light perception, then I would say no. I'd rather I would undergo treatment to just maintain it. Yes.
And Professor McDonald, is that your experience speaking to other patients as well?
Oh yes, many of the patients would accept a clinical trial that maintain the visual function that they have, either acuity or field. To get the benefit of improvement, that's astounding. And you can't bottle their enthusiasm. It's not going to fit in the bottle. It's effervescent.
And maybe to kind of pivot a little bit, but also, I think it ties into the clinical trials discussion. And we're obviously having a discussion about data, maintaining vision. And, I think some of this discussion is easier to follow for people that may be familiar, and harder to follow for people that may not have a full understanding of how vision works and clinical trials work. So one question I wanted to ask to both Francesca and Dr. McDonald really speaks to language and communicating with patients when you're discussing both a diagnosis, a clinical treatment, and then potential clinical trials. And so maybe Francesca, I'll start with you. I think you have the benefit of also being a researcher, so you have sort of certain sophistication with and familiarity with a lot of the medical terminology. But maybe from your perspective, what kind of language should be avoided for people when they're either getting diagnosed or learning about clinical trials when they're having a discussion with their healthcare professional?
Well, I've worked on informed consents. I've written material for the public, and I believe that there's nothing that should be omitted. It's just maybe the way of saying it that should be a little different. But of course, I mean, the language should be such that everybody can understand. Usually, someone speaks about a level like a 12-year-old should understand. Anyway, it's important that every person with any background should be able to understand, because we don't know who these patients are, and citizens too in general. And I don't think there should be anything that should be really avoided, because all the concepts must be there, and we maybe avoid the acronyms or use them, but in parentheses always put the explanation and the definition and what it is. But that's it.
I think that's good insight. And Dr. McDonald, maybe for you, how do you adjust your language when speaking with a patient versus maybe speaking to Dr. Girach about a treatment?
Well, speaking to a colleague, you can use the full vocabulary, but speaking with a patient, I usually start with right to the start. I talk about their expectations, because then that frames the conversation, and also their knowledge base. Francesca talked about the consent document, because this is a clinical trial with a procedure, and in seeing a physician or an optometrist or other eye care provider, when you walk in the door, consent is assumed that you're going to share information. But once you go into a clinical trial, there needs to be a written document with risks and benefits that are itemized, and in relaying those, there's the written document. Maybe the patient can't see the document very well or can't read it at all, so you have to really orally go through it. And it has to be, in my environment, somewhere around grade 8, so that the patient has a realistic understanding of what the risks and benefits might be. I think I should stop there, because I could go on, and that's the time.
Well, I just wanted to follow on. Sorry, go ahead, Dr. McDonald.
Well, one thing I think is very important is that the patient has somebody else in the room. So there's another, as they say, set of ears that can that the two can. What actually happened? What did you understand? I'll stop there.
Yeah, I agree. I think, just another perspective, and like you said, Dr. McDonald, someone else to say, did you understand the same thing that I did? And I think for someone to have someone else to talk to about, is this the right thing for me? Someone that knows that person's personal situation, I think is just really valuable as well. So that's a great insight. And just wanted to kind of follow up to that, maybe talking about placebo or sham procedures. Do people express concerns about getting a sham or placebo versus an active drug? And maybe Dr. McDonald, I'll start with you, and then Francesca, I'll ask you a similar question.
Well, yes, I think in my experience, they would be concerned about getting the placebo because they're recognizing that it's another opportunity lost in their case. But you can see that the design of this particular trial is a period of non-treatment, which you could replace the word placebo as being no treatment being provided with a sham injection, for example. But that information is going to be very helpful, because that individual could potentially then proceed into the next part of the trial, and using the information generated from the observation over a year with what is the observation over the next year. The before and after is genuinely important data. I do know that there is this placebo effect, and the regulators really want to have hard data, and so the placebo effect is there, but the hard data, the scientific data, can trump that and show there's a definite change in between treatment and the observation.
And Francesca, maybe I'll just ask you from a patient perspective, the concerns related to placebo or sham, and kind of any insights that you have from a patient perspective.
Yes, I totally agree with what Dr. McDonald said. And I would add that also, besides perceiving it as a waste of time, it can be a burden for patients. Mm-hmm. So patients need to travel, sometimes need to undergo multiple tests, which sometimes are really, can really bother, can really be annoying. For example, in my personal experience, it's very hard for me to undergo tests because I can't keep my eyes fixed. And that's the same for most blind people, many blind people. So, and if you're talking about sham, it's even worse. I mean, you have to use anesthetics and you have to miss out days of work. And it's a lot that you have to go through. And for patients, it can really be stressful. And they might think, okay, I'm wasting my time. I'm not my time, but I'm wasting time that I could be receiving when I could be receiving the treatment. And the placebo effect is rationally, I do understand it, and I know that it's there, and it's actually wonderful because it shows us that the power of the mind is very big. I mean, it can help us heal. But I also think that in some cases where, for example, you don't see anything except for light, and you have objective measures, outcome measures, then I find it very hard to come to terms with it. And when I was screened for the Illuminate trial, and I really hoped to get in, and if I had been found eligible, I would have participated. But it was really hard for me to come to terms with it, to know that I need to receive placebo for a year. And I think that all efforts should be made to find alternative designs, because it's really, I mean, one thing is if we're talking about food supplement, people take vitamins and they say, oh, I feel better today. That's might be the placebo effect. But when you have no vision and then you start reading letters, I mean, I know perfectly why the sham procedure is used, but I think that effort should be made to minimize that.
Yeah.
Francesca, I was just going to take the unenviable task of trying to represent the regulators here and suggest that of course, the idea of the regulators insisting on us as a company to have sham-controlled trials, especially in the phase 3 trial setting, is to protect the patients in order to ensure that we have genuinely a drug that gets out there that's approved, that's both safe and has benefit. And so it's really to reduce the amount of bias that's there. And as much as it's a nuisance, it's difficult, more painful, troublesome for us also to incorporate sham patients into their trial. It is probably a necessary evil for us to make sure that we do the trials and that the integrity of the trial is done in the right way to maintain the integrity of the data that comes out, and that all of us should find out for sure whether a drug is safe and beneficial for patients ultimately.
And maybe I'll just conclude that with saying, I think it's really important for us as a company and for healthcare providers to remind ourselves about the burden that patients are under to participate in clinical trials and the sacrifices that patients are making to participate, and also to be held accountable to make it as easy as possible. So while we obviously can't eliminate placebo, I think there are many things that we can do to make the experience as seamless as possible. And, that's why, Francesca, you are part of the ProQR patient and caregiver steering committee, exactly to hold us accountable to say, this is our experience and this is how you need to make it easier for us. So I think, it's always good to be reminded of the burden for the patient, because I think we can always do more and need to be held accountable to do that. So thank you for sharing that, Francesca. So we have 5 minutes left, and a comment has come in and a question has come in that I want to get to, and a comment from our colleagues in Brazil, which I think is just an interesting comment on the communication. And it reads, communication is complex in the Usher group, so Usher syndrome, sometimes having to translate English into Portuguese and then into sign language. So it's a good example of the challenges across IRD. So I think that is a great example. Obviously we communicate in English, and when you're communicating something 3 times English into a second language, into sign language, is obviously presents a lot of challenges in how you communicate information accurately. And the last question that has come in, is it too early to determine if RNA therapy has limits on the ability to revive older retina cells sucked by an IRD? Or is this a patient-by-patient basis? So maybe Dr. Girach, I'll hand it to you, and then maybe if Dr. McDonald has any thoughts.
Yeah, I think it's a similar answer to a previous question that was asked. I think it's early days at this stage. We have preliminary data that shows very promising improvements in vision overall, both in best corrected visual acuity and also the sensitivity test that we revealed the data on. So it's early days. I think we're seeing some useful results across the board. The improvements that we saw in the graphs that I shared were mean improvements across the entire cohort of patients that were treated. And therefore, if we look at that target registration dose, there was approximately 9.5 lines improvement in vision in the treated eye, compared to a nominal amount only in the untreated eye. And that's a mean improvement in vision. A change from baseline. So that suggests that across the board, overall, there seems to be an improvement that occurs. But of course, we have examples where the vision did not change, however, other parameters changed and improved. So it can differ between patient and patient, and it depends also, I think, on the degree of advancement and the stage at which the patient is at with regards to the disease itself. And of course, the earlier you can catch that, the better the theory is that we should be able to at least either halt the disease or improve the disease with any drug that could be potentially improving this. But I think this is quite an important question. And I don't know, Professor McDonald, if you have any other insights which could help here also.
I think you said it quite well and aptly. The issue is that perhaps the younger individual may benefit more. The older individual that had more damage, let's say, may not benefit as much. And there may be some that benefit greatly and some that benefit not very much, let's say, according to the data, the hard data. But the patient perspective here is very important. And it may be that they say, well, yes, the data doesn't show there's an improvement, but I feel, regardless of placebo effect, that there has been a benefit for me, and over the long term, that will probably play out. I'm just saying it in another way. One other thing to consider is that within a clinical trial, we can't use all the sophisticated tools that we have, but there are some very sophisticated tools that we can probably use in the future to look at the photoreceptors to look at them in detail with imaging to see what changes. And so that's for the future.
Great, thank you. And I want to thank you all and for participating, in particular, Dr. McDonald and Francesca. It's really been an honor for us, and I know kind of for everyone listening, just learning more, connecting more, and building this community. You both play an essential part, and we're really grateful for your time today. So thank you for participating. And with that, I'll hand it over to Dr. Girach for the closing remarks.
Yes, thanks, Jessica. And, really, my warm thanks to Professor McDonald and also Francesca Diodati for their participation here. And indeed, the participation for all of the people who dialed in and who logged on, and all of your questions as well. It's great to know that you're very warmly engaged and actually thinking about inherited retinal diseases and, sharing with the journey that we're all on together in order to find meaningful treatments for patients. Ultimately, there is still a lot of uncertainty related to COVID-19, but really our current plan is that we'd like to meet again as part of this meeting, the EFF meeting in 2021, hopefully in Amsterdam. But it's likely to be a hybrid of both a virtual meeting as well as an in-person event. And of course, if it's in-person and if the current limitations are still there, it will definitely be a socially distanced in-person meeting. We hope that all of this settles in time for us to re-engage as we were before, and those are very fond times that we all remember, of course. But we will share more details with yourself later on this year. Providing you with more information. Just also a big plug, and maybe if I can move to my last slide there, a big plug for our trials that are enrolling at the moment, in particular the Illuminate study, which is the LCA 10 study. We have, so you can find out more information from our website, ProQR.com. We have a dedicated page, web page dedicated to Illuminate study itself called lcatrial.com. And you can find out more information about the trial itself. You can see at the ProQR.com website details of the other two studies, the Aurora study and the Stella study, which are also recruiting at this stage in different diseases. And of course, you can also find out more information about the MyRetina registry and tracker through either the Foundation Fighting Blindness website or indeed our website, where there's a link to that particular connection to our website. And then, of course, all together with regards to the Eye on the Future Forum, you can see the website given on the slide there. Please visit us and all of the other different social media forums. I'd love to hear from you and stay engaged and also stay connected. And we thank you once again, and we look forward to re-engaging again, hopefully early next year in a much more normal setting, we hope. So thank you very much and have a good day.
Thank you, everybody.
Thank you very much.
Have a good day.